Hi, I'm Dr. Kim Anderson with the Miami Project, and I'm going to talk to you today about the results of our subacute spinal cord injury Schwann cell clinical trial that we've been conducting for a few years now. So, what do we talk about when we are talking about repairing the spinal cord, and how is that relevant to this clinical trial? Well, you know that there are many different things that happen inside the spinal cord after an injury. So there are many different repair strategies that we need to employ. One of those repair strategies is to try to prevent the secondary damage from occurring. But this needs to happen early after injury, and these are more our neuroprotective types of interventions. Another thing that we need to do is try to neutralize the inhibitory scar tissue that ends up developing around the lesion site, which prevents regeneration from occurring. That's a separate repair strategy. A third repair strategy is actually inducing regeneration of axons that have been severed or damaged. And you can see some of those in damaged axons here. And then finally, there are cell replacement repair strategies. And Schwann cells are one type of that cell replacement strategy. And the goal is to try to replace the cells that form myelin around the axons and to provide a repair component in that regard. So let me tell you a little bit more about Schwann cells. Well, first of all, what are Schwann cells? Schwann cells are a type of cell that is in the nervous system, and it wraps around axons, individual nerve fibers, and creates what we call myelin sheath. And the myelin sheath you can think of as insulation around the axon that enables electricity to be conducted for neural communication. Schwann cells are normally wrapped around all of the nerve fibers in your peripheral nervous system. So that's everything in your arms, your legs, your chest, everywhere except for your brain and your spinal cord, because the brain and the spinal cord are the central nervous system. So the Schwann cells normally live in the peripheral nervous system. So why would we want to use Schwann cells to try to repair the central nervous system, spinal cord? Well, we know from many, many years of research that Schwann cells are responsible for promoting regeneration of axons that are damaged in the peripheral nervous system. We also know that Schwann cells produce many different growth factors and extracellular matrix components that they can release and provide to the environment that they are around. We know that if Schwann cells are transplanted into the spinal cord or into the central nervous system, they can form myelin and ensheath axons in the central nervous system. We also know that those axons that get the Schwann cell myelinating them can restore conduction. We can test this by electrical physiology. We also know that after a spinal cord injury, Schwann cells naturally enter the spinal cord in substantial numbers. So what we're trying to do is augment an already occurring process. Schwann cells are easy to access. We can get them by taking out a sample of a peripheral nerve without any significant injury. And very importantly, Schwann cells can be transplanted autologously. That means that they can get them from your own body and then transplant them back into your own body. And therefore, people are not required to have immunosuppression medication. Finally, we can obtain them in very large numbers. And importantly, for the future, Schwann cells can be genetically engineered. So we may be able to, for example, program a growth factor to be released in significant amounts by Schwann cells, which may then enable Schwann cells to have on additional activity. So for the subacute Schwann cell clinical trial, it's been a very long process to get to its completion. As you can see, we started seriously thinking about the clinical trial in 2008. And we had some preliminary discussions with the FDA to determine what we needed from our pre-existing research to generate new data in order to enable us to go forward into humans. And so the FDA informed us that we needed to get some good manufacturing process and manufacturing procedures for human Schwann cells so that they could be transplanted into people. We needed to conduct some toxicology and tumorigenicity experiments. And we also needed to do some additional toxicology in our pig model to really evaluate the safe dose and volume for the clinical protocol. So we did all that between 2008 and 2011, and in late 2011 we were able to submit the investigational new drug application to the FDA, and they had some questions, and finally in July of 2012 we were enabled to move forward with our clinical trial. So in November of 2012 we actually initiated the first participant, and from there it took us 2.5 years to transplant all 6 participants in our trial. And I'm going to tell you about the data for all of those individuals. So I mentioned that this is a subacute clinical trial, and it is a Phase I trial. So a Phase I trial is entirely evaluating safety. And so the design of the trial is quite conservative so that we do not put any people at unnecessary risk. So the individuals that qualified for this clinical trial had to have a thoracic level injury between the levels of T3 and T11. They had to be Asia Impairment Scale Grade A, which is what we call complete. They had to be between 18 and 60 years of age. Initially, we had the cutoff at age 50, but then we changed it mid-trial up to 60. And individuals had to be within 30 days of their injury in order for us to do the peripheral nerve harvest. Initially, we had set this limitation at 5 days post-injury. This was very difficult to get individuals to our center. We then changed it to 7 days and finally to 30 days post-injury. Now, these are just some of the exclusion criteria. There were several more. Individuals with penetrating injuries were not allowed, or complete spinal cord transection. If the lesion was too low in the spinal cord and involved the conus, that was an exclusion. If somebody had significant multi-system trauma or a closed head injury, that was also exclusionary criteria. Or if there was very significant severe neuropathic pain at the admission of the trial. So a diagram of what the overall trial is demonstrated here. So we take a sample of the sural nerve, which is a peripheral nerve in the leg. That's for sensation only. And we take about 10 to 20 centimeters of that out of the leg, and we put it into the cell culture dish. The average for all 6 individuals that we transplanted was about 16 centimeters in length that we took. So that nerve then goes into a culture dish in our cell processing facility, and it takes about 3 to 5 weeks to process the cells so that we get a pure population of Schwann cells and they meet all of our different exclusion and release criteria. Now that process for the cell processing is very regulated. It's very important that we know what the cell composition is and that it is safe to put into human beings. So this is an outline of the process. It takes about 3 to 5 weeks for human Schwann cells. There's a little bit of variability between people. But before the transplant, the cells have to meet our release criteria. And those involve sterility, endotoxin levels, mycoplasma has to be negative, purity has to be greater than 90% for Schwann cells, and the viability has to be greater than 80%. If the cells did not meet these criteria, they were not able to be transplanted. So then, schematically, we would go back and do a separate surgery on the spinal cord. This is approximately 2 months after the actual injury has occurred. And this diagram shows the injury, and you can see that there's a lesion area, but there's also tissue around the lesion that may have spared nerve fibers, but they've lost their myelin. And so the cells are injected into the center of the injury, and the goal is that they will stay around there, and also that some of the Schwann cells will form myelin around the injury-spared axons that have lost their myelin, and try to create remyelinated and supportive environment for improved connections. So the timeline of the trial involved many different aspects. Initially, you have the spinal cord injury. A significant amount of screening has to occur before the nerve harvest can occur. And then the individual goes through their regular rehabilitation while their cells are being processed. Remember, this was early after injury. Once the cells are almost ready to be transplanted, we then go through some baseline assessments to determine the level that the individual is at prior to the transplantation. Then we have the transplant. We do assessments for the first couple of weeks after injury, and then at 2 months, 6 months, and 12 months after the transplant. And then there is a long-term protocol that individuals go into, which follows them for an additional 4 years. So the entire time that we are following individuals is actually 5 years from the time of transplant. So remember I said that this is a safety trial. So our primary outcomes are safety. The most important safety assessments that we have are looking at compliance and the actual feasibility of the trial, evaluating the adverse events and see if there are any adverse events that are related to the actual Schwann cells or to the surgery, and then looking at the stability of each individual's neurologic level. We don't want to make anybody neurologically worse. We also have some secondary safety assessments, and that is to look at the spinal cord via MRI and make sure that there are no mass lesions that have developed. And also we want to evaluate pain and spasticity to make sure that there is not a worsening than what is already the natural occurrence that happens after spinal cord injury. And then we're also looking at several exploratory measures, and those are functional independence measures for disability in general and for spinal cord injury specifically, looking at electrophysiology measures of subclinical connections, looking at autonomic changes, and then a global impression of change of that each person's perception. So for enrollment, we screened 39 individuals, and we actually enrolled 9 of those, and 6 people were transplanted. 1 person that was enrolled was not transplanted because that person self-withdrew before the nerve harvest actually occurred. The second person that was not transplanted had a nerve harvest, but the cells became contaminated and were not able to transplant that. And the third person that was enrolled but not transplanted had the nerve harvest but converted to an incomplete status before the transplant, so we were not able to transplant him and we had to exclude him. So this table here shows demographic information from the 6 individuals that were transplanted. You can see that they were all men, and they had injuries from either motor vehicle, ATV, or motorcycle accidents, with one exception of a diving. And here you can see their single neurologic level of injury. You'll notice that one individual had a T1 injury, and I mentioned that our inclusion criteria was from T3 to T11. This T1 was due to one sensory abnormality on the ASIA evaluation. The lesion via his MRI was at T3, so we requested an exception from the Institutional Review Board prior to doing the transplant to be able to keep him in the study. Everybody started out as AISA, and we had a variety of backgrounds of individuals. So this is information about the transplant and about the cells. This table up here shows for each individual the number of days from their spinal cord injury to their nerve harvest, and the number of days from the nerve harvest to the transplant. And you can see the average number of days between spinal cord injury and nerve harvest was 14. And the average number of days between nerve harvest and transplant was 26. We also have information about the dose. So this was a dose escalation trial. The first 2 individuals received 5 million cells in a single injection of 50 microliters. The second 2 individuals received 10 million cells in a single injection of 100 microliters. And the final 2 individuals received 15 million cells in a single injection of 150 microliters. The duration of the injection was approximately an average of about 4 to 4.5 minutes for each individual, and at a rate of 22 microliters per minute. Now we also have information about the purity of each cell population, which you can see for each individual here. On average, it was about 96.9% purity for Schwann cells. And here we have an image of the final products for each of the persons that was transplanted. In green are all the Schwann cells. We also have an example, this is an example of ultrasound that is done during surgery so we can see and identify the injury epicenter. This is the syringe positioning device, which is what mounts to the operating room table, holds the syringe that is then used to inject the cells into the spinal cord. And here's an example of the needle of the syringe that is going into the spinal cord for 2 different individuals during injections. So importantly, we were looking at adverse events. And this slide shows you 3 different ways that we are evaluating our adverse events. Here in the top graph, we are looking at the cumulative number of adverse events based on different body systems, and it is for all 6 subjects over the entire 12 months that each one was evaluated. And so the most common adverse event is urinary tract infections, UTI, skin issues, and a few pain issues. These are all things that you would expect to happen after spinal cord injury in the first year. What we have in the second graph is also all of the adverse events per subject, but this time it's presented across each month of follow-up. So you can see that there's quite a few number of adverse events that are happening before the transplant even occurs, which is what you would expect. And then the gray hashed line here between 1 and 2 months post-transplant is representative of when 5 of the 6 individuals were discharged from their inpatient rehabilitation. And you can see that after discharge, many of the adverse events start decreasing. There was one individual that I will get to in just a second that had significant amount of trauma associated with his injury, so he was not discharged from rehab until 9 months post-transplant. And then finally down here, we have adverse events per dose, per subject. So here's subject 1, 2, 3, 4, 5, 6. And the black bar represents the number of adverse events that subject had pre-transplant. The gray shaded bar is the number that they had within 1 month of the transplant. And then the hashed bar is the cumulative number they had between 2 and 12 months after the transplant. So we had a total of 53 adverse events for all 6 subjects over the entire 12 months they were evaluated. You'll notice that 21 of those were for subject TMP-008, which was our 5th transplant. This is the individual that had a significant amount of trauma associated with his injury, and those adverse events were due to that trauma. There was one serious adverse event, and it was deemed to be not related to the Schwann cells. It was related to the spinal cord injury. Very importantly, there were no deep vein thromboses or no pulmonary emboli. There were no meningitis infections, and there are no pseudomeningoceles that developed. These are very important adverse events that did not occur. And there was one altered sensation in one subject that occurred 3 months post-injury, post-transplant. And it was determined by the Data Safety Monitoring Board that that was not unexpected for spinal cord injury. And I'll show you a little bit more about that adverse event on the next slide. So here we are presenting neurologic data, which is the INSCI data, International Standards Neurologic Classification of Spinal Cord Injury. Many people call this ASIA data. But what you'll see for each here, green indicates normal segments, red indicates where their sensation is absent, so it's 0, and orange indicates where there is sensation, but it's not normal and it's not absent, so it's altered. And so let's start with the first subject that was transplanted. You can see that this orange segment at T2 is where he had the adverse event that he got an altered sensation. What happened was 3 months after the transplant, he started feeling a hyposensitive area underneath his armpits on each side. This corresponds to dermatome T2, and that therefore raised his single neurologic level up to T1. But by 12 months post-transplant, this had resolved back to normal on one side, but was still altered on the other side. The second individual did not have any change in his sensation at his spinal cord injury level, but you will notice that he's got some altered activity in his arms. And this individual actually had peripheral nerve damage as a result of his spinal cord trauma, or the trauma that he sustained as well. And that is something that was persistent for him, but unrelated to the spinal cord injury and unrelated to the cells. The third individual that we transplanted actually gained one level down in his sensory, and this is called in his zone of partial preservation, or the ZPP. The fourth person that we transplanted had his ZPP go down one level as well. The person that had the most additional trauma associated with him, his single neurologic level actually went up because of an altered sensation in one dermatome on one side, but his ZPP actually went down one level. So that was a gain. And then the final transplanted individual is the one that had the most improvement. This individual gained 9 sensory points, and he converted to an AISB at 6 months, and this was persistent at 12 months. So AISB is indicative when you gain sensory function down at the most sacral levels of the cord. However, with all of these gains, his single neurologic level actually stayed the same. So this really shows how important it is to look not just at INSCI levels or ASIA grades, but you need to really get into the detailed data from the evaluation. Now we're looking at the imaging data via MRI, and what you see here is the baseline MRI for each individual. Remember, their baseline was done right before the transplant, so this is approximately somewhere between 4 to 6 weeks post-injury for each individual. And then we also have their imaging at 12 months post-transplant down here. Now remember, we did not have a lesion size restriction in this trial for entry into the trial. So we have pretty significant lesions, which you can see down here in the table. We've got lesion length and lesion volume at baseline and at month 12. So we have some pretty significant lesions. They ranged in length from 2.5 centimeters to 7.6 centimeters, and they ranged in volume from 0.8 cc's up to 4.1 cc's. Now what we did notice when we went back and had our neuroradiologist calculate the lesion length and volume at baseline at month 12 is that there was a significant reduction in the lesion length and in the lesion volume at month 12 post-transplant, which is indicated here at the end of the table for an average for the group. Now, what we don't know is whether or not this reduction is any relationship to the Schwann cells or if it's a consequence of atrophy that naturally occurs to the spinal cord after the injury. Next, we have our important evaluation of pain. This is something very, very important because we don't want to make anybody's pain get worse. So what this diagram is showing you is for each individual, we've got a diagram of their body and their pain at baseline and then at month 12. So we've got subject 1, 2, 4, 6, 8, and 9. And what the red color indicates is a neuropathic pain. The blue color indicates a musculoskeletal pain. And the purple color over here indicates a mixture of neuropathic and musculoskeletal gains, which is not uncommon to see after spinal cord injury. So we had 3 individuals, which are indicated by the blue stars, that never had neuropathic pain prior to the transplant and did not develop neuropathic pain within the 12 months after the transplant. We had 2 individuals with the red star here that did have neuropathic pain prior to the transplant, and that neuropathic pain was persistent out to month 12 post-transplant. And then we had 1 individual, the individual that had the most trauma, that had pain pre-transplant, but it was a little bit difficult to identify what that was. It mostly had musculoskeletal pain qualities, as you can see up here in the diagram. And then by month 12 post-transplant, he did develop neuropathic pain. So that shows that 50% of our participants had neuropathic pain that developed within that first year of spinal cord injury. However, prospective and longitudinal data that is out there in the literature indicates that actually about 60% of people with spinal cord injury will develop neuropathic pain during their first year. So our data fits with the expected data for neuropathic pain development just related to spinal cord injury itself. So what this tells us is that we actually cannot say whether or not Schwann cells are going to have a relationship to neuropathic pain because we only have 6 individuals. However, in these 6 individuals, there is not a worsening or an attenuation of neuropathic pain in relation to the transplant. And then finally, we have some exploratory outcomes that we wanted to look at. This table has several different things on it. ISS stands for Injury Severity Score, which is a measure of the amount of additional trauma the person sustained in addition to their spinal cord injury. FIM is the Functional Independence Measure, and SCIM is a Spinal Cord Independence Measure. These are 2 measures of overall ability, functional ability. MEPs are motor evoked potentials, and EMGs are for electrophysiological assessments that we did on different individuals. And then PGIC is the Patient Global Impression of Change. This is their feeling of how their change was after the trial. So first thing, let's talk about injury severity and its relationship to function. There were 3 individuals that had significant trauma that did contribute to impaired function in the beginning. Individual right here had an injury severity score of 20 and had sustained a rotator cuff injury with his spinal cord injury. So this individual had lower than expected FIM and SCIM scores at baseline. Remember, baseline is at least 1 month post-injury, but recovered as would be expected by month 12. Subject number 2 had the brachial plexus lesion, which did impair his functional ability initially, but that individual also recovered back to an expected level by month 12. And individual number TMP008 is a person that had significant chest trauma and femur fracture related to his initial injury. And he had an injury severity score of 34, which is pretty significant. He had very low functional mobility scores at baseline, and they were still below average by month 12. But this individual had complications related to a femur fracture that prevented him from starting rehabilitation until approximately 9 months post-transplant. And so his recovery is slower than expected by month 12. If we look at subclinical connectivity via electrophysiological data, the person TMP009 that also converted to an AIS-B also gained some electrophysiological activity in his legs. So that's below his level of injury. And then there were 3 individuals that gained EMG activity in their legs by month 12 post-transplant. And then the global impression of change, everybody felt that they had gained something by being in the trial. And the closer to 1 that the number is, the more positive that they felt that that gain was. So what can we conclude about this trial? Number 1, we can conclude that it was safe. And it appears to be feasible to do this type of a nerve harvest and an additional spinal surgery in the subacute setting. If it's a single center trial, it's better and more feasible to have a 30-day window in order to have individuals stabilize and get them transferred to the facility if that's required. Doing the surgery and implanting the Schwann cells did not appear to impede any kind of natural recovery, though if a person has a severe injury trauma associated with their spinal cord injury, that may impact their participation in rehabilitation, which may impact their rate of natural recovery. But that doesn't seem to have a relationship to the Schwann cells. And finally, there was not an attenuation or an exacerbation of neuropathic pain by doing the surgery or the transplant. So some questions that remain: what about incomplete injuries? What about smaller lesions? What about larger doses? And what about sustained rehabilitation? These are all questions that we have incorporated into our other Phase I clinical trial that is currently ongoing, which is transplanting Schwann cells into individuals with chronic spinal cord injury. And we're not going to discuss that in this presentation. So finally, it's very important that we acknowledge everybody that was involved in this trial. So first of all, Dr. Alan Levy and Jim Guest are principal investigators on the trial, and Dr. Dalton Dietrich is the sponsor of the IND. Mary Bunge, Damian Pierce, and Pat Wood have been very involved in the preclinical and basic science assessment and the cell processing development and guidance as we go through the clinical trial. Aisha Khan and her cell processing team, Adriana Books, Maxwell Donaldson, and Gagini Athada, have been very critical in the actual cell processing, development of the protocol, and actual execution of the cell processing for each individual. Marina Diditsi was the primary clinical coordinator for the subacute clinical trial, and Katie Gant, Katie Nagel, and George Mshelishvili have also been involved at different aspects. Dr. Eva Widerstrom was critical to the pain evaluations, and her team with Jim Adcock, Jackie Tibbett, and Jessica Cambridge. These are very, very important to our evaluation of the safety. Dr. Rosie Curiel and Dr. Mario Olivaria and Marianne Rosado were the psychologists that were involved in the trial. I didn't get into the psychological data, but we were very involved pre-transplant and post-transplant out to 12 months to ensure that individuals really understood what they were being involved with for a Phase I safety trial. Dr. Green and Dr. Cardenas and Dr. Dalal, Dr. Vani, Dr. Wang, Ellie Balawis and Shelley Garcia were all heavily involved with our Jackson counterpart and enabling the inpatient procedures to occur. And Dr. Alberto Martinez Arozala is the medical monitor for the trial, so helping us evaluate and interpret the data. And Dr. Lloyd Zucker was the site PI at Delray Medical Center, which was a screening site for us, who contributed one subject, but that was that subject that became incomplete prior to the transplantation and didn't get transplanted. And Dr. Efrat Saraf Lavi, who was our neuroradiologist who helped us evaluate the lesions, and Francisco Benavides and Andrea Santamaria, who helped Dr. Guest with the electrophysiology. And finally, Danielle Sully and Letitia Fisher helped with many parts of the regulatory aspects of the trial. So funding, this was funded largely, very largely, in part by donations to the Bonacani Fund to Cure Paralysis, as well as donations from the Brian Reich Paralysis Foundation. So as you can see, this has been a significant amount of effort to conduct this clinical trial. It's taken quite a large team of individuals, and it has been successful. So if you would like to know more about the clinical trial or ongoing clinical trials, don't hesitate to contact us at the Miami Project.